Thank you very much. Actually, I realize that I'm the last speaker, so it's the only thing that prevents all of you going to have some fun in New York, so I'll try to do this as quick as possible. So basically, something that was discussed yesterday is how we can develop better clinical trials and better clinical development approaches for patients in colorectal cancer, now that we know that there are different subtypes of patients with, at least with advanced disease. So basically, these are my disclosures. So if we look at the way that we have been developing drugs for colorectal cancer, actually, this is a summary slide that shows that we have been doing quite well in terms of improving overall survival, although that we could argue that this clinical development has been done in a very empirical way. Only retrospective analysis have actually provided us opportunities for predictive biomarkers, including KRAS and NRAS mutations. So hopefully we will be able to do a little bit better in the next future. So this is the outline of what I'm going to cover. Actually, we have done I would say a deep work on providing important information on mechanisms of primary and secondary resistance for EGFR inhibitors, and we'll touch this a little bit more. Also, we know that there are a few groups of patients, unfortunately very infrequent, but that have very innovative and we could say even very active treatments, like the BRAF mutant population, the HER2 amplified population, even the mutant population, and the MSI, and as it was shown yesterday. But also there are some patients that have translocations that have opportunities with clinical trials that are being conducted right now. But the fact is that probably the data that has been generated for subtyping colorectal cancer based on these multi-omics or transcriptomic approaches, actually, I'm going to show you how we can provide new insights for new clinical trials. So basically, in looking at the EGFR pathway, we all know that this is a pathway that it's important in colorectal cancer. EGFR is overexpressed in almost all patients with metastatic disease, and there are at least two monoclonal antibodies, cetuximab and panitumumab, approved for the treatment of patients with metastatic colorectal cancer. However, there are important downstream effectors that are frequently mutated, even constitutively mutated in patients with colorectal cancer, like KRAS, NRAS, and BRAF. So if we were going to treat a patient with an EGFR inhibitor at first line as a single agent, this is something that we are not going to do. But if we were in the conditions of treating a patient with an EGFR inhibitor, actually only around 15% of them would have an objective response. And the reason for that is that there are, as mentioned, important aberrations and irregulations in patients with colorectal cancer that prevent the activity of these compounds, at least as single agents. This includes, as mentioned, KRAS mutations, NRAS, BRAF mutations, also some patients that have combinations of these mutations and PIK3CA mutations. Also, de novo HER2 amplification and c-MET amplification. This is a little bit more rare, but still there are patients, around 3 to 4% of them, where we can find these amplifications from the early beginning. The seminal work that has been done with cetuximab and panitumumab has provided us, this important information on how we can select the patients for being treated with these compounds in the first-line setting. And as you can see in this slide, I think that it's important to say that by decreasing the population, almost half of the patients that from the intention to treat population, actually we have been able to decrease the hazard ratio for overall survival from 0.93 to 0.69. It's a major advantage. Nevertheless, if we treat patients in this way, NRAS, KRAS, wild type, with an EGFR inhibitor in combination with chemotherapy, we also know that we are going to be selecting mutant clones or amplified clones that are there from the early beginning. And this is what is being shown in this slide. So basically, at the beginning, we cannot find these mutant or aberrated clones. But at the time that we put pressure to the tumor over time, actually we are selecting these clones that I did mention before. And this is what we are going to be observing at the time that the patient clinically progresses. progress. So this is something that has been very nicely addressed by several groups. And at the time that we try to evaluate which are the mechanisms of primary resistance and secondary resistance for EGFR inhibitors, actually we can see that the scope of aberrations or the regulations that we have at the time that the patients develop secondary resistance, it's completely different, or a little bit different at least, from those that develop primary resistance from the early beginning. So we understand that the scope of these molecular aberrations evolves over time, something that I mentioned before in my first presentation on the dynamics of the disease. And even we know that there are patients that develop EGFR mutations in the ectodomain of the receptors. This is something that has been really very well described for colorectal cancer, and actually there are generations of new monoclonal antibodies, basically combinations of different antibodies like the Symphogen 04 compound or the Merrimack 151 that are active in these patients that have developed these ectodomain mutations. And actually there are several clinical trials at this time point, well, not several, at least 2, that are evaluating the activity of these compounds in patients that have responded to a conventional EGFR inhibitor and have developed secondary resistance based on mutations at the ectodomain of the receptor. So this is one particular aspect. So for patients that are being treated with EGFR inhibitors, we are understanding which are the mechanisms of secondary resistance, and therefore we are able to promote clinical trials that try to abrogate these mechanisms of secondary resistance. As mentioned before, there are a few populations of metastatic colorectal cancer that have a different behavior. Yesterday we discussed a lot on the BRAF mutant population, and we know that these patients have mainly a poor prognosis when they are metastatic, and mainly these patients do not respond to conventional EGFR inhibition. So the rationale for this population of patients was, at the beginning, to treat them like the melanoma patients that bear BRAF V600E mutations. But you know that treating these patients with single-agent BRAF V600E inhibitors didn't work at all. And at least 2 labs at the same time elucidated which were the mechanisms of primary resistance in this case, because the patients didn't benefit from the early beginning, to BRAF inhibition. And very nicely, these 2 groups elucidated that there was an immediate upregulation of EGFR that was not amplified, was not even overexpressed, but at the time that you treat the tumors with a BRAF inhibitor, there was an immediate feedback loop that promoted overexpression of a receptor. And at the time that in these preclinical models, both in vitro and in vivo, these tumors or these cells were treated with a combination of an EGFR inhibitor plus a BRAF inhibitor, the results were completely different. And you can see that there was complete inhibition in this long-term clonocyte test, as well as in this in vivo model. So basically, this has promoted the development of several clinical trials evaluating combinations of EGFR inhibitors plus BRAF inhibitors, or even with a third drug like a PI3K inhibitor or a MEK inhibitor. The bottom line here is that there is right now mature information that at least one third of the population, 30% of the patients, have objective responses to this approach of combining an EGFR inhibitor with a BRAF inhibitor. So another population that was presented last year at the ASCO meeting was the HER2 amplified population. This is a rare population, as mentioned before, in naive patients, but if we look at patients that have progressed to EGFR inhibitors, actually this population may grow up to 10% of the patients. The important thing here is that in this beautiful academic trial, the Heracles trial, the combination of trastuzumab and lapatinib actually induced an objective response in around 1/3 of the patients, and control of the disease with a median time to progression of around 5.5 months. In a very refractory population. So all these patients had progressed to all available treatment options. This is as frequent as 10% of the patients, 12% of the patients, if they have been previously treated with EGFR inhibitors. A more rare event actually shows that some of the patients, very few patients actually, have HER2 mutations, also in colorectal cancer. And this is data that was generated at Boston and also at Memorial Hospital in looking at the frequency of these mutations. The important thing here is that if you find in any of these multiplex test mutations for HER2 in colorectal cancer, actually there is strong preclinical data showing activity in these cells, in these tumors, with a combination of trastuzumab or plus neratinib, or even with neratinib as a single agent. And right now, at least 2 different clinical trials for this very rare but still present population are evaluating these compounds in the clinical setting. We can skip PD-L1, obviously, for the MSI patients. Yesterday we saw the data, but this is also quite infrequent population in metastatic colorectal cancer. And the final point that I wanted to make on these rare populations comes from the observation that some patients with metastatic colorectal cancer can have also kinase fusions or translocations that produce aberrant proteins, mainly ALK, BRAF, and TRK fusions in patients with colorectal cancer. And also, at least 2 different clinical trials are evaluating TRK inhibitors in patients with colorectal cancer. And, there are case reports showing that these patients can benefit from the treatment. So basically, this is what we have for these very rare populations. But if you count all the patients that have a druggable, actionable aberration in patients with colorectal cancer, is not as high. As I did mention before in gastric cancer, it's almost the same. Around 30% of the patients at maximum have clear dependence on a particular aberration. So what are we going to do for the other set of patients? Naturally, several efforts from independent groups have tried to provide characterization of colorectal cancer in different subtypes based on this multi-omics approach, including gene expression. So if you look at the data that was presented up to 2 years ago, you can see that there were several different signatures, some of them dividing colorectal cancer in 3 subtypes, some other in 6 subtypes. So it seemed to us that this would be really a nightmare, and we didn't figure out which was the right one. So basically, these are the caveats of these signatures that had been published before, mainly, different subtypes. And one important caveat of all these series, most of them, actually almost all patients have stage 2 and stage 3. Very few patients have stage 4. But the most important thing is that it could seem that actually this data is really very discordant. But when we look at detail, actually you will see that this is not the case. And an important effort that was actually led by Sage Bionetworks, by the group in Seattle, actually collated all these different gene expression signature series in trying to provide one single and well-defined and validated classification. And this was what was done and presented initially at ASCO 2014, and now this was published at the end of last year, basically showing that we can define at least 4 different populations with colorectal cancer. And if you look at the data, basically these are the different signatures and publications that have been presented before. And at the end, you can see by the colors that actually all these patients, almost all of them, collate very well in these 4 different types. And the names are basically expressed here. So the first group is the CMS1. This is the microsatellite unstable group, accounting around 14% of the patients. The second one is what we call epithelial canonical, and the name of canonical basically comes because these patients have deep and profound activation of the Wnt and the MYC pathway. The third one, and this actually, the canonical accounts for 37% of the patients, so this is the most frequent one. The third one is what we call epithelial metabolic, because all these patients have important metabolic features. This accounts for 13%, and finally the mesenchymal subgroup number 4, accounting for 23% of the patients. So when we look at the clinical characteristics of these patients, actually we can see that for the 3 subgroups, CMS 2, 3, and 4, they are more frequent in the male population as expected, but the CMS 1, the MSI, actually it's more frequent in the female population. Actually, you can see also that there are differences in the proportion of the different subtypes depending on the location of the colorectal cancer. What about the genomic characteristics of these subgroups? Well, as you can see, the CMS1, as expected, we discussed this before and yesterday, this is an hypermutated population. So these are the patients that mainly, not all, but mainly have mismatch repair deficiency. So we expect that these patients have several mutations based on the dysfunction of the mechanisms of DNA repair. But the others basically have the opposite. So the other subgroups actually have chromosome instability, and as you can see in this slide. And as expected, this also correlates with the MSI status and the CM cluster status. Regarding BRAF mutations, as expected, they are more frequent in the CMS1 subgroup. KRAS mutations are equally present in all the subgroups except for the CMS3 or metacolic group subgroup, which actually around 70% of the patients have KRAS mutations. One important thing here in this multi-omic approach is that these samples were not only evaluated for the gene aberrations. There was a profound effort to look also at the mRNA level and at the protein level, whether there were different features between patients in one subgroup and the other. And as you can see, for example, in this mRNA enrichment analysis for the signatures, you can see, for example, that the CMS2 subgroup, the one that it's called epithelial canonical, actually, as mentioned before, it has upregulation or activation of Wnt targets and MYC targets as expected. However, the mesenchymal one, for example, we know that these tumors actually bear an EMT properties, so we can expect that they will have activation or overactivation of mesenchymal properties and also TGF-beta and matrix remodeling activation. And what about the immune? Basically, we see this in the CMS1 population, the MSI, and unfortunately, this is something that we have always mentioned in patients with colorectal cancer, why these compounds, PD-1, PD-L1 inhibitors do not work in the others, basically because the T-cell infiltration, it's not really very apparent. Although some new data will elucidate which are the patients that may even benefit from these immune treatment options. And the same relates for proteins. So I'm not going to go in detail, but you can see that the gene set mRNA analysis correlates very well with the proteomic analysis. So the bottom line here is that the information is really very robust at the genomic DNA level, at the mRNA level, and at the proteomic level. Finally, this classification provides also prognostic information, and you can see that the mesenchymal subgroup actually has the worst overall survival and disease-free survival. Remind that these patients mainly are stage 2 and stage 3. But at the time that these patients relapse, actually the things change a little bit. And as we mentioned also yesterday, those patients that have CMS1 that relapse, this is not a very frequent situation, but those that relapse actually have a worse prognosis once they relapse. So this is a summary of the clinical, genomic, proteomic, and phenotypical properties of these groups and also the location. But the important thing here is, are going to be these CMS subtypes predictive for any particular treatment? Obviously, for the CMS1 population, This is immediate. We know that this population of patients actually are those that benefit from immune checkpoint inhibitors, and also around 50% of the patients have BRAF mutations, so they are going to benefit from BRAF inhibitors combined with EGFR inhibitors. What about the other subtypes, and which kind of information actually can provide this characterization? Well, actually, it can provide a lot. So, for example, if we go back to the CAMBATA-4 data that was generated in patients with metastatic colorectal cancer that were refractory to conventional treatments and were treated with cetuximab as a single agent. Actually, this was one of the studies that defined that the KRAS mutations were negative predictors of activity for cetuximab. You are in a situation doing this gene expression profiling where you can characterize which patients benefit more and less. Even all of them, the patients that have been evaluated here, are RAS wild type, BRAF wild type. So all these patients should have benefit from the treatment, and we know that all patients do not benefit from the treatment. But in here, you can see that those that are canonical, so CMS2, are actually those that benefit from the treatment. However, the mesenchymal ones, even they are RAS wild type, BRAF wild type, do not benefit at all from the treatment with cetuximab. So you can see that even with a retrospective evaluation of the data of clinical trials that have been published several years ago, we can even elucidate a little bit more which patients are going to benefit more or less from the treatments that we have right now. And actually, in thinking on the way that we could do new clinical trials, let's focus right now, for example, on the RAS, so KRAS and RAS BRAF wild type population. Mainly, these are the patients that I mentioned are CMS2 or CMS4. For the CMS2 population, we know that these patients are those that are more sensitive to EGFR inhibitors with the study that I mentioned before. But at the time that these patients progress, mainly these patients are going to have amplification or overexpression of further tyrosine kinase receptors. So potential combinations of these inhibitors inhibiting these other receptors like MET, like HER2, may work in combination with cetuximab. On the contrary, for the mesenchymal, we know that these patients do not respond to EGFR inhibitors. So in principle here, we should combine these compounds with other agents that target the aberrations that these patients have. For example, TGF overexpression, MET overexpression, and also MEK overexpression. So this relates, for example, to this particular population. Let's think about the RAS mutated. Basically, the RAS mutated, most of them are CMS3. So this is the epithelial metabolic. And we know that in this particular population, actually, the tumor cells or colorectal cancer tumor cells and models that we have, PDX models, combinations of pan-RAF inhibitors or MEK inhibitors with metabolic enzyme inhibitors show preclinical activity. So this is something that we could do in this particular population. I mentioned before that, unfortunately, for the colorectal cancer setting, very few, beside the MSI subgroup, so the CMS1, very few patients actually have lymphocyte infiltrates in the metastatic disease. This is not universal, obviously for CMS1 we have mentioned before, but actually for the mesenchymal population, there are some kind of T cells infiltrates, but mainly, or some of them actually have Tregs instead of cytotoxic T cells in the tumor. So, as an example, one preclinical approach actually showed that if in this particular population of patients you combine a TGF-beta inhibitor with an immune checkpoint inhibitor, you may see activity, at least in this preclinical model. So this is something that we could explore in the clinics. What about the CMS1 group, what we call immune? Most of the patients, as mentioned, are MSI, and we know that these patients actually benefit from PD-L1 inhibition. But there are some patients that have hypermutated tumors, and the mechanism this high load of mutations is not related to mismatch repair deficiency. And actually, we know that for this particular population of patients, PD-L1 blockage may also work in this setting. And which are these patients? So, well, there are several groups of patients, but some of them actually, the cause of hypermutation may be related, for example, to POL-E mutations that are well defined in other tumors like endometrial tumors, but also in colorectal cancer. So something that we should characterize also to better understand which is the population of patients that are hypermutated but not related to mismatch repair deficiency. And as mentioned in the previous preclinical model for the mesenchymal subgroup, actually we may combine immune stimulatory drugs also with inhibitors of the immune suppression like the TGF-beta pathway, what actually mentioned. Finally, also some data that was presented at the last ASCO meeting in 2015. So this was a retrospective evaluation of patients included in the core study, the treatment with regorafenib against placebo. You know that a total of around 750 patients were included in this particular trial, but we were able to collect tumor samples from around 1/3 of the population. So we can always discuss which is the real mechanism of action of regorafenib, but at least we know that this compound has important anti-angiogenic properties, including obviously others, but basically it's a BGFR inhibitor, a PDGFR inhibitor at least. So basically, in looking back to the gene signatures of these patients, these 281 patients, actually we were able to generate an hypothesis that those patients that had tumors that were more angiogenic dependent, like the CMS4, were the patients that actually benefit more from the treatment with regorafenib. And actually, if you look at the hazard ratios for overall survival, this subgroup, the EMT-CMS4, is the one that had the better hazard ratio for overall survival. And as expected, the MSI population didn't benefit at all. And surprisingly, also the CMS3 population, the population that is metabolic, didn't benefit from the treatment. I would also caution about the numbers. So this is results from one retrospective evaluation of 1/3 of the population that was included in the correct study. But for me, the important thing here is that this is a model of how we can go back to all the clinical trials that we have presented so far, in which we have available tumor samples, and try to look whether we can separate which patients benefit more or less from the treatment. So in summary, very similar to the summary that I presented before for gastric cancer, with this characterization that we are doing in clinical trials and in academic centers, also with molecular profiling, actually we have generated data that around 1/3 of the patients have aberrations that are druggable, like EGFR overexpression or amplification, like HER2 amplification. Basically, we have seen that for this population, especially the quadruple wild type, the BRAF mutant population, and the HER2 amplified or mutant population, actually there is activity with abrogation of these deregulated oncogenes, and also, for example, for the TRK translocations. But for most of the patients, actually, this is not going to work. And in the way that we have been doing for the colorectal cancer subtype, at least we have been able to generate these 4 different biologically distinct subtypes that are enriched for key clinical characteristics, but also, more importantly, for molecular alterations and pathway traits that provide not only prognostic information, but as I try to show you, they may provide also predictive information for the drugs that we have in the armamentarium right now, and also provide important information for new clinical trials based on these hypothesis generating data. But again, importantly, think always on the dynamics of the disease, because basically these tumors under stress of not only of EGFR inhibitors but all other compounds may change over time the proportion of cells and the traits of the different cells, the different populations that compose the whole tumor burden. Thank you very much for your attention.